Good morning, everyone. First, I would like to thank the organizers for giving me the opportunity to speak and tell about my work. Uh, so basically, I work at the interface of physics and biology, and try to take in the physical principles and try to understand how biological systems work. And particularly work on signaling networks. I hope so. This is like a diverse audience is there, so everyone knows what a biological cell is, right? So yes, we are all made up of those cells, and so basically my work is to kind of understand how these cells are able to kind of perceive their environment, take in the information, gather the information from the environment, and process it, and eventually make decisions, important decisions, like whether to divide or not, or whether to differentiate, and so on. So before I just start, what I wanted to show was this video. It's a very famous video. It will what you would expect to see from here is that this is actually a white blood cell. It's like it will be chasing a pathogen over here, and eventually it will track this pathogen, and eventually at some point of time it will consume it. So when we first we'll be looking at this video, and you will see that somewhere there's these cells have some eyes or something, but actually in reality they don't have these eyes as we do. We can see people and follow them or whatever. So but cells they have a more rudimentary kind of mechanisms to kind of sense their environment. What they have eventually is these receptors, okay, and what the receptors what they do is that they sense this chemical signals, right? Which can be any molecules or something like that, and when they let's say when a molecule kind of when any of the molecule binds to these receptors, it triggers it sends us information downstream, okay, and inside the cell you will have these different other molecules would be there, okay, of different types, which are the protein molecules, and they would be so once it gets the signal from the membrane, it kind of will transfer that information. It will activate some protein, okay, and that protein will again activate some other protein. In this way it will pass down the information downstream and to the nucleus, where let's say some proteins will be produced, right, and eventually the cells would respond to that, right? And so it is very important to understand the network which is there, which is the bridging this the outer the membrane to the inner nucleus, and because any defects in inside this this network can lead to a lot of pathological consequences it can have, such as cancer and all. So a lot of important processing that the cells can do, okay, and one of the important processing that the cells can do is be sensitive to any change in the chemical stimulus, okay, in the environment, right? Yeah, so I just like to compare this with how a camera works, the sensitivity part. So suppose most of you have some experience using a camera. So suppose you have an object, and it's a very dull object. It's there, not much you can get out of that object, and now you want to, when you are taking the pictures, say so you want your camera to be very sensitive, in some sense, so that any change, any difference in the illumination can be enhanced. And so the cameras, they have these sensors, right, and based on that, when it gets the light from that object, so whenever any light which is light which is slightly greater in luminosity, which enters the sensors, so it gets enhanced, and in that sense you can get a sort of a bright image in that sense. So And similarly, to this, you can have like a cell also working in that same fashion. But here, the object is basically, what I say like is in chemical in nature. Okay, so here it has to sense any slight change in the chemical the stimulus it has. So you can have a very dull profile, right? So this H stands for high and L stands for low. So the cell has this task to kind of sense this subtle difference in the intensity of this chemical signal. And so what it has is basically this, as I said, in the network side, this cascade, signaling cascade it has, which is basically a series of molecules where each molecule is activating the other molecule and that molecule activating the other molecule. So whenever any it gets the signal, so it kind of amplifies the signal in that some sense. And as I said, it has like this kind of an input-output profile, which is like similar to that of a like sensor, and it's able to kind of enhance any change which is there in the chemical stimulus, so that at the output, like it will have like a high contrast between the between the low and the high signal over here. So so this allows the cells to kind of make like right decisions, right? Because from the input side, it's very much not very clear like how much high and low are the signals are there. But when you have too much, like having sensitivity, high sensitivity is also not very good, okay, because in the environment that the cells they work in, it's very noisy, I mean in some sense. So your signal is always would be like corrupted with some noise in that sense. So when you and just compare it with photography, like when let's say you want to like take a picture, okay, which is in a very low light condition, but when your camera has like too much high sensitivity, then you can get this graininess in your picture, right? It's just because of that there is this in the low light condition you have these the light signals that you are getting are very random in nature, and also the camera the sensors are also kind of gets some way itself like gets corrupted. So but in a very, when it has very low sensitivity, then also like you will get a very dull kind of a picture in that some sense. So this is the same thing that happens in a cell, okay. So when so cells like they get like a noisy signal which is from the output, okay, from the input, sorry, and also inside the cells, like because like these this interaction, the molecular interaction that is happening, these are like random in nature, okay, and this can induce noise into the system. And adding all these things, so basically like what would happen is that if you have too much high sensitivity, then the output that you will be getting, obviously it will have like a good contrast, like it will have like good contrast between the low and the high input, output. But the fact is that you can also get wrong signals, the spurious signals also, because this noise can drive your output to in a wrong regime in that sense. Right. So the question, that we are trying to ask over here, is that so basically the sensitivity it depends on the system that I am working on, the signaling cascade. It depends on the number of layers of interaction that the molecules have, right? So there can be 2 layers of interaction, okay. So once the signal is received from the input, so it gets passed on to, let's say, the one molecule, and that molecule then again interacts, like activates the other molecule. So there can be 2 layers of interaction can be there, and but this will be like not much sensitive. But then even you have like more layers, like when you have like, let's say, the ground figure which you are seeing, let's say there are four layers are there. So there you can have like a very high sensitivity in the response. But the fact is that like but you have this noise also in this in your system. So that what would it do is that it will corrupt your signals. So basically when you have a high sensitivity, you're getting a sharp output, but your system, I mean the output may not be accurate. I mean sometimes it may be accurate, sometimes it may not. But when you have like a low sensitivity, your the output is accurate, okay, but the response, the output that you are getting is very dull. It will not be very much magnified as such. So it will be difficult for the cells to kind of distinguish between what is the let's say a low signal and what is the high signal level. So the work that I'm trying to do over here is that trying to understand whether there is some trade-off is there or not, okay, between like let's say how accurate the signal, I mean the output can be, and also how much sensitivity is optimal for a cell to work with, okay. Yeah, so yes, that's it. Thank you.
Thanks, Soumyadeep.